Well, hey, AP Biology. Hope you are doing well when you watch this. So we're going to walk through cellular respiration together, fermentation, metabolism, and these are pathways that harvest and store chemical energy. All right, so moving on. Heterotrophic organisms, that's us. We have to eat, right? Hetero, different, some troph, eat. So we have to eat something different. Must capture energy from their environment, of course, to maintain life. So that's related, big idea. To common to all are core processes that move energy and matter through living organisms or systems and thereby support life. So that's, of course, common. When we see the word common, we should think conserved or evolution. So I would pause the screen right here and see if you can do all of this. For time purposes, I'm moving forward. All right, so puffins. They are like the cutest birds ever. Actually, my daughter loves puffins. They're in Noelle on Disney Plus. I'm sure that doesn't matter right now. But anyway, so yeah, example question from another textbook I have. How does food, like the small fish, power the work of life? And so you can see right here, there's a picture in your notes. I want you to fill it out. So we'll just move these out of the way. So we start with light energy. Of course, this is photosynthesis. We're making organic molecules in O2. Okay. And then that happens in photosynthesis, of course, in chloroplast. Knowing the anatomy of the chloroplast, that'd be good. But those organic molecules and oxygen move to cellular respiration in mitochondria. Okay? Plants do both. Don't forget that. Plants do both. Common misconception, even in higher levels of thinking. And then cellular respiration in mitochondria, CO2 and H2O. So these processes are related. And of course, we expel heat at the end. We generate ATP. We don't create it. Again, mitochondria, the powerhouse of the cell, right? Well, don't think powerhouse means it's creating energy, laws of thermodynamics. So we're converting chemical energy, organic molecules, into cellular energy, ATP, and of course releasing heat as we go as a byproduct, okay? So catabolic pathways break down, we're breaking down things, catabolic. And so the breakdown of organic molecules is exergonic, which means energy is exiting. Fermentation is a partial degradation of sugars that occurs without oxygen. So big key right there is without oxygen. Aerobic respiration consumes organic molecules in O2, okay? And so, yeah, we're taking what we build in photosynthesis, or what we eat, of course, if we're not plants, and the oxygen that we make or breathe in, or, take in, and we are trying to, that's our goal, right? We're trying to make as much ATP as possible, all right? So let me take the writing off. So here's the equation. Knowing this would be super important. Here's a balanced equation, ATP and heat. And so although carbohydrates, fats, and proteins are all consumed as fuel, we're just going to kind of start with glucose. But yeah, so let's say someone's on keto diet, and they're not eating many carbohydrates. Well, that still could be funneled in the system. I'll show you that down the road, okay? So cellular respiration. It's a process with production of ATP. It's an aerobic process, which means we need oxygen, okay? And so it's breaking down glucose to CO2 and H2O, okay? Energy is extracted from the glucose molecule, released in a stepwise, step-by-step process, okay? Again, oil rig, don't forget that. Oxidation reduction enzymes, oil. Let me see if I can write with a mouse. I need to get a writing pad. I know I say that like every time, but it's a thing. Oil rig, oxidation reductions still are happening here. Coenzymes, such as so on and so forth, okay? So you can see right there, there's our good old oxidation. Oxidation is loss. Reduction is gain. Sorry, that's kind of like messed up. Like, that's weird. Taking time on YouTube to try to figure this out. Oh, that is weird. Maybe like extend it this way. Ah, that's better. So yeah, it's a little messed up, but you can see glucose. We're oxidizing that. We're reducing oxygen into water, but we're yielding all that energy, that ATP energy. Okay. So you can see right here, it happens in the glorious mitochondria. During cellular respiration, fuel is oxidized and oxygen is reduced. Organic molecules with an abundance of hydrogen, right? That's our fuel. Carbs, fats. Excellent fuel. That's why you pasta load if you run or do really long endurance sports. Pasta loading is important with that. As hydrogen is transferred to oxygen, energy is released that can be used to an ATP synthesis. Okay, so cellular respiration, they're broken down. Electrons from organic compounds are usually first transferred to NAD+, a coenzyme, right? So it accepts an electron, much like NADP+ in photosynthesis, and it functions as an oxidizing agent, which means it is reduced. Okay, so each NADH represents stored energy that's tapped to synthesize ATP. Okay, so enzymes called dehydrogenases remove electrons and hydrogens, dehydrogen, right, without hydrogen, forming NADH. So one hydrogen is released in the process. Okay, so this is kind of what it looks like again. Glucose is being oxidized. Oxidation is loss, and oxygen is reduced. But the goal, again, the goal is right there. That's a really horrible circle. All right, and all this NAD plus and NADH. Well, here's what it looks like. You do not have to know this. Of course, this is like biochemistry stuff, but nonetheless, hydrogen's coming from food. We reduce that, and it goes to the electron transport chain to help us generate ATP. And so that's what we just talked about right there. So electrons transfer to oxygen, stepwise process, okay? So they are passed, right? It's a chain. And so we'll see this down the road a little bit. And so electron transfer to oxygen. So oxygen has to be waiting. Oxygen is the final electron acceptor. And the energy yielded is used to generate ATP. Okay, so where am I at? Nope, I'm not there. Pause, wait for it. There we go. And so it kind of looks like this, right? We could burn, like you could burn sugar, or a gasoline, truck. There's some videos that's horrible accidents where gasoline trucks explode. They, all that energy is released as heat and light. But if you do it as a stepwise process, you can kind of mine it. You can mine that fuel, mine that energy. And so here's just another, small energy, right? Kind of mine it versus large activation exploding, and that's not going to work, okay? So there's two enzymes in cellular respiration, NAD plus and FAD, okay? It's a coenzyme, which means it's necessary for things to work correctly. NAD plus molecules used over and over and over again. And so this, again, oxidation reduction reactions, very important. Cellular respiration includes 4 phases. So yeah, four different phases. You need to know where each one happens, what happens in each one. So glycolysis, lysis, the breaking down of sugar, glyco. Okay, there it is right there, break down glucose. Does not use oxygen, so this is where fermentation is going to come about here in a little bit. Occurs in the cytoplasm. The prep reaction, or the link reaction, or pyruvate oxidation, is pyruvates oxidized. So these two pyruvates are oxidized. They enter into the mitochondria. Electron energy is stored. Again, we're taking the energy from food and trying to convert that into cellular energy, ATP. So it's stored in NADHs, and CO2 is released. Okay. Then we have step 3, good old Krebs cycle, citric acid cycle, the first intermediate, but occurs in the matrix of the mitochondria. I'll show you a picture in a second. In the series of reaction that releases 4 carbons of CO2. Again, there's 6 carbons to be released, right? C6H12O6. And so it turns twice per glucose molecule, produces 2 immediate, again, that's the goal. So some of these, glycolysis, Krebs cycle, they're making a little bit of ATP, but they're not making enough to maintain homeostasis. Where does that happen? Right here. This is where all the ATP is made. I mean, the majority, not all, majority. And so the electron transport chain extracts energy from the reduced coenzymes, passes electrons from higher to lower states. Thus, through the electron transport chain and chemiosmosis or oxidative phosphorylation, 32 to 34 molecules of ATP happens in the inner membrane. So let's look at this picture right here. A couple of pictures here. So here's glycolysis. Again, sorry for the wording, SE, but here it happens in the cytoplasm. If oxygen is around, preparatory reaction. So those pyruvates happen inside the mitochondria. And then the Krebs cycle happens within the matrix, which is the very innermost part of the mitochondrion, and then the ETC over here, okay? So again, net gains are always kind of important to understand. You got 2 here, 32, 34. There's a difference there because it takes energy to pump this stuff out, so that's where the difference comes from. That's kind of where we're looking at. 4 different phases of cellular respiration. Each phase is important to think about what's happening in it, where it's happening, and what's happening, okay? So that's kind of what we're going to go through, okay? So catabolism, for the breakdown of glucose under aerobic conditions, occurs in 3 linked biochemical pathways, right? The complete breakdown. Glycolysis, converted to this molecule called pyruvate, two pyruvates actually. Pyruvate oxidation, which is oxidized to acetyl-CoA and CO2. And then the citric acid cycle, also known as the Krebs cycle, which is oxidized further to CO2. Okay, so it kind of looks like this in a diagram form. Being able to like draw this and like explain it would be an amazing study tool, study habit, note card, something like that. Like glucose, breaking it down to pyruvate, happens to the cytoplasm. If O2 is around, those pyruvates go into the mitochondrial matrix, where they're further changed, you know. And then those products are shuttled into the citric acid cycle. And then those products from the citric acid cycle go into the inner mitochondrial membrane, where we make all, again the goal, ATP. Okay? And so, kind of looks like this. This is another great picture to study, to be able to explain out loud, to understand what's happening. Cell-CoA being made in pyruvate oxidation, funneled to the citric acid cycle. Electrons carried via NADH and FADH2. Again, that's where we're storing the energy from food into oxidative phosphorylation. Okay? So in eukaryotic cells, the inner mitochondrial membrane is a side of electron transport chain. And another process called chemiosmosis, which is where hydrogens are flowing downhill, fueling ATP synthase, together making up oxidative phosphorylation. Okay, a smaller amount of ATP is formed in substrate level phosphorylation, which you might be like, what in the world? What are the differences between? I mean, phosphorylation is adding a phosphate. I get that, you know, teach. But what's the difference between substrate level and oxidative? Well, let me show you. Here's substrate level. Oxidative involves chemiosmosis, ATP synthase, electron transport chain. Substrate level is when you have your substrate, ADP, you take the phosphate from the substrate and directly add it to the ADP, thus creating ATP, and enzymes do that. And so that's just an example there, okay? You can work through these questions right here on the next slide. I'll be going through these in class, right, because we're trying something different out with cellular respiration to see if it works a little bit better for everybody. So go ahead and pause, work through these questions out, and we'll keep going, okay? All right, continuing on. Outside of mitochondria, let's start with glycolysis, all right? Sugar splitting, sugar breaking, lysis burst, okay? So we're breaking down sugar, okay? There's an investment step and then a harvesting step. What's great, if you have the mind to invest, is that investing, of course, brings, hopefully, returns, right, when you invest. So we have to give a little ATP at the start of this to activate it, to harvest more ATP. So there's an investment step, okay? So 2 ATP are invested, and then we're harvesting, so 4 ATP are gained, which of course that means a net of 2 ATP in total, right here. Good old math. Okay, so what does this kind of look like? Oh, let's take off the writing. Okay, so here we go. There's 10 reactions. You do not have to know all the products and the steps. That's not the goal of this course. But the final products would be helpful to kind of know. 2 molecules of pyruvate, 2 molecules of ATP, and 2 molecules of NADH, which is electron carrier, right? Reduced oil rig. And so, investment, payoff is what happens, okay? Forming 4, so we got 2 NADHs, 2 pyruvates. So this is what it kind of looks like. You take glucose, breaking it down to these intermediates. You don't have to know these steps, right? Okay, 2 molecules, splits into 2. Multiple different steps. Again, each step is catalyzed by a different enzyme. There's enzymes for all of this. And then finally you have 2 molecules of pyruvate. All right? So you can see right here. And so this is just an example of a reaction that occurs repeatedly, right? Step 6, dehydrogenase, kinase. We know what kinases do, right? Adding a phosphate. And so you have glycerolide 3-phosphate dehydrogenase, 6 and 7. Those are just 2 examples of what's happening in this process, okay? So oxidation reduction is exergonic. So this phosphate is oxidized. The energy is trapped via reduction in NAD+, NADH. There you go, right? Where'd the hydrogen go? It came from here to there. So this is just kind of how it works. Oil rig, oxidation reduction reactions. And then Substrate level phosphorylation, here's step 7. We have 2 phosphate, phosphate, and then the phosphate is transferred to ATP. Again, if you need to review oxidation reduction or substrate level phosphorylation, there you go. Okay, so I would pause here and answer this question. During the redox reaction and glycolysis, which one of the molecules acts as the oxidizing agent? Which one acts as the reducing agent? All right, moving on to section 3. So pyruvate, right? Where does pyruvate come from? When you're kind of studying these things, think about, you can ask these questions. Okay, pyruvate, 2 pyruvates come from glucose, breakdown, glycolysis, right? And so it's a pivotal metabolite. If oxygen is not available, keyword, of course, not, fermentation occurs in a cytoplasm. And anaerobic, which means it doesn't need oxygen, okay? This is the foundation for those who like to run or sprint. You can jog longer than you can sprint, dead sprint. It's all about ATP, fermentation. Can't make enough ATP to keep generating, your ability to keep those muscles contracting, using up all that oxygen. So during fermentation, glucose is incompletely metabolized to lactate or to CO2 and alcohol. Of course, lactic acid happens in us. And so, yeah, that's this burning, possibly this burning sensation. Some scientists debate that or not. But when you work out for a long time, if oxygen is available, pyruvate enters the mitochondria for aerobic respiration. So what's happening in fermentation? Okay. NADH is reoxidized by fermentation. There are many different types of fermentation, but all operate. Key, very big theme here. Let me see if I can do something different than pink. Is their ability to like highlight? Yes, there we go. That was not a good choice. All operate to regenerate NAD+, okay? Because you have to keep that going. You have to keep NAD+ going, coming back at least, right? Okay, the overall yield is only 2, right? So again, if you think back to our first lesson, the first couple minutes ago, we're making 32 to 34 in the ETC. We're only making 2 here. Sprint versus jog, you can jog a lot longer because you're replenishing that oxygen debt. So the end product is lactic acid or lactate. Okay, so NADH is used to reduce pyruvate to lactic acid, regenerating NAD+. So it occurs in microorganisms and complex organisms, including us, including vertebrate muscle during exercise when oxygen cannot be delivered fast enough. Okay, lactic acid, if you like lactic acid, here's some great examples of some products you might enjoy, right? Cheese, yogurt, sauerkraut. I think there's a teacher at our school that absolutely despises sauerkraut, but nonetheless, other bacteria produce chemicals anaerobically, including isopropanol butyric acid, propionic acid, and acetic acid. Acetic acid is vinegar. So examples of that sort of stuff, balsamic vinegar. I mean, there you go, right? So lactic acid, a lot of good food comes, or recipes come because lactic acid happens. So what exactly is happening here? Well, this, again, don't have to know the molecular forms. The names will be important, but definitely knowing what's happening here is very, very important. So what is happening? All right, so let's walk through this. Glycolysis, glucose, breaking down. NADH, pyruvate. Oxygen's not around. So lactate dehydrogenase takes pyruvate and converts it into lactic acid. Okay? But what's important here is NAD+ is regenerated, which allows this process to keep going. If NAD+ is not regenerated, this process shuts down. Okay, so you have to regenerate NAD+. You have to oxidize NADH. Okay, that is lactic acid fermentation. Okay, alcoholic fermentation, ethyl alcohol, ethanol. So again, NADH is used to reduce acetaldehyde to ethanol, regenerating, again the goal, regenerating NAD+. Yeast, some plant cells under certain under anaerobic conditions. Breads, alcoholic drinks. Bread rises. Why? CO2. Champagne CO2 is trapped. That's why you get the fizzing when you open the bottle. And so glycolysis, NADH, pyruvate, different enzymes. There's our CO2 that makes bread fluffy. There's I remember all the time growing up in Auburndale, we would drive by Nature's Bakery, like factory, and it literally smelled so good because you smell the alcohol burning off, but the CO2 is what makes your bread fluffy. And so with all the air holes in it, you have acetaldehyde and then alcohol dehydrogenase making ethanol. But why is that important? Because we're regenerating NAD plus again. Okay, and so here's just an example of rate of fermentation, type of sugar. This is for yeast. Just some example data here. Like, what's the data saying? Okay, kind of think about what's working there in the process. Okay, so advantages, yeah, quick burst of ATP. Disadvantages, lactate and alcohol are toxic to cells. Lactate changes pHs, causing muscles to fatigue. Again, we're trying to pay off that oxygen debt, right? We use a lot more oxygen than we have, so we got to pay that off. That's why you breathe so hard after like sprinting or hard working out. And so efficiency, 2 ATP produced per glucose, 14.6 kilocalories. But look at this, complete oxidation of glucose can yield up to 686 kilocalories. You can do the percent there to figure out it's not very efficient, right? But it is a process that is necessary because sometimes we just don't have enough oxygen around to keep functioning, keep homeostasis going. So here's your question for this section. Why is replenishing NAD plus crucial to fermentation? Go ahead and pause this and answer that. We'll talk about it in class. So moving on to step 2. We already talked about glycolysis. We talked about when oxygen is not around, it goes into fermentation. Now, the next step, step 2, pyruvate oxidation. And step 3 is the Krebs. So pyruvate oxidation, this is when oxygen is present. It occurs in the mitochondria and eukaryotes. Products, CO2, acetate. Acetate is then bound to coenzyme A, acetyl-CoA. NAD plus is reduced to NADH. Okay? So in eukaryotic cells, if oxygen is present, pyruvate enters the mitochondrion to complete the full oxidation of glucose. So again, 2 pyruvates are made from glycolysis, so you make 2 NADHs from the process. Okay, so here's what it looks like. Again, enzymes are carrying this out. You don't have to know those names. 2 molecules, pyruvate oxidation, only if oxygen is present. Okay, cleave off another carbon, add coenzyme A, we reduce NAD+, and then we form 2 molecules of acetyl-CoA. Okay, that is pyruvate. Here's just another picture of it to help you kind of see what's happening here. See one of the carbons is missing at acetyl-CoA. And then where is it happening? In the matrix. Pyruvate oxidation is also called the PrEP reaction or the link reaction. Either way, I would know all those synonyms, okay, just to be prepared. And so then you have, again, this is a transmission electron microscope, color's been added. Matrix, crystalline folds, intermembrane space, inner membrane, outer membrane. Here's our good old mitochondria, the powerhouse of the cell. The Arnold Schwarzenegger. Y'all probably don't even know who he is. That's maybe. Anyways, and so citric acid cycle. So step 3, we go into the Krebs cycle, okay? So it completes the breakdown of pyruvate to CO2. Each turn of the cycle oxidizes a molecule derived from 1 pyruvate. So the cycle turns twice then. Why? Because 2 pyruvate entered the mitochondria when oxygen was around. So we generate 2 ATP, 2 NADHs, 2 FADH2s, just another coenzyme reduced per glucose molecule, okay? It's called the citric acid cycle because that's the first intermediate you'll see here in a second. But there's 8 different reactions. You don't have to know the molecule. You don't, again, it's beyond the scope of this course to know the molecular formulas and all the enzyme names and all the names. But in general, knowing where it's happening is always important. Mitochondrial matrix. Knowing what the goal is always important. What's the goal? We have CO2, okay, being released. There's the 2 CO2s, right? Boom, boom. Okay, 2 carbons, I mean. Make it NADH. Make it NADH. GTP, which is form of ATP. FADH. Those are our goals, right? Those are our goals. And so just seeing a little biochemistry, here's what's happening. So there's citrate or citric acid. That's the intermediate. So this molecule is waiting on it. Again, this brings up some great conversations about Darwinian evolution and irreducible complexity and all that. Then you make all these different molecules. Again, you do not have to know the names of these. Yes, there might be some connect questions about them, but again, we practice harder than we play, right? So redox reactions, ATP formation through a GTP kind of reaction, substrate level, and then redox reaction. So here's your question. In the citric acid cycle shown in figure 711, what molecules capture energy from the redox reaction? Okay, and then how is ATP produced? That's in your book. Also, you can scroll up and look at the picture, the 4 pictures I just went through. And then what processes in your cells produce the CO2 that you exhale? So you've been breathing in a lot, maybe stressful breathing when watching this video. And if you're still watching it, hey, good for you. Okay, that's amazing. What processes release the CO2 you've been breathing the entire time? All right, here we go. The last step of glycolysis, the electron transport chain. So, I wish my buttons worked. Oh, my keyboard the way they're supposed to. Okay, here we go. Sorry, talking to myself. Oxidative phosphorylation, NADH oxidation, oil rig, is used to actively transport protons across the intermitochondrial membrane, resulting in a proton gradient. Again, we're building a proton dam. Diffusion of protons back across the membrane drives to the synthesis of ATP. Okay, so here's kind of the reaction. It's ETC, electron transport chain, respiratory chain, embedded in the inner membrane. Where is it at? Prokaryotes, the plasma membrane. Of course, they don't have organelles, membrane-bound organelles. And so this is what it looks like. Being able to draw this, again, just using shapes. All these shapes, these proteins have names. ATP synthase is the most important one to know. Really the only one to know for the exam in May. But nonetheless, the energy that we stored from glucose, from food, we reduce this, right? We use that energy to actively pump. So again, this is active transport. Actively pump protons across the membrane. This looks a lot like photosynthesis, right? The electron transport chain photosynthesis. And so you can see here, oxygen is a final electron acceptor. If you're thinking of this as a conveyor belt, a machine, you have to have something waiting at the end for the electron. If it doesn't, if that's not around, the entire thing breaks down. Okay, and so you can see we're building a dam, a hydrogen ion dam up here, a gradient, a lot on one side, not much on the other. And again, we allow those hydrogens to flow through an opening, through a little tube. And ATP synthase, again, ATP synthase is way more complicated than this oval you see. It's like 13 different parts, give or take. And we're producing ATP through oxidative phosphorylation and chemiosmosis, okay? Lots of different videos. I'll watch them. You can watch this one. I think there's some on my north side. So again, active transport, we're creating a hydrogen ion dam. Oxidation is always coupled with reduction, right? So we're oxidizing NADH. We have to reduce something. And so what do we reduce? Oxygen gas, the final electron acceptor, making water, which we either use or excrete as waste. And so ATP synthase uses the hydrogen ion gradient to drive the synthesis of it through chemiosmosis. We've learned this term before, but let's learn it again, or let's review it again. Movement of ions from higher to lower, okay? That's what it is. And so that literally turns ATP synthase like a motor, and it converts the potential energy of proton gradient into chemical energy in ATP, again. So we're converting energy here. And so it kind of looks like this, has multiple different units. And so oxidation, create the gradient, pump through it, and then we use that chemiosmosis to oxidatively phosphorylate ADP and inorganic phosphate. So here's a question for you. Tragic, right? This was a picture, I believe, multiple years ago when I was teaching in Orlando. A lady in Orlando, I didn't know her, but she was on the news. Power went out from a hurricane. I forgot which one. But she literally put a generator inside her home, and she placed paper towels, not paper towels, but towels. She knew that the gas fumes are dangerous. So what she did was she lined the door with like dry-off towels, like bathroom towels. Rolled them up, put them along the door, thinking, okay, that's not going to let any gas come through, but it did. And so tragically, her and her daughter died. So carbon monoxide binds strongly to hemoglobin, thus blocking oxygen binding. Predict what the effect on electron flow and ATP synthesis would be if the oxygen concentration inside the mitochondria was decreased. Okay, a good, action verb there. Predict, right? So pause it now. Figure out your answer to that. We'll talk about it in class. So chemiosmosis relies on concepts: diffusion, of course, plasma membrane, and the phospholipid bilayer, and then nonpolar interior. Protons have a charge. It's all about charges, rather they can't cross. So here's just another picture from a little bit of a dated textbook, but you can see oxidation of NADH, oxidation of FADH2 drives the formation of the hydrogen ion dam. Okay, creating that gradient. Diffusion can't go through everywhere, but there is an opening to go through here through ATP synthase, and therefore that motion is turned into powering the motor, making ATP from ADP and inorganic phosphate. Okay, so ATP, yeah, it's a very complex molecule, but similar. ATP is similar in all organisms. Again, when you see this phrase right here, think evolution, common descent. Okay, the proton gradient is set up across the cell membrane. Chemiosmosis happens between those specialized organelles and eukaryotes. So it's similar in almost all forms of life. Again, big idea one, evolution, okay? And so chemiosmosis can be demonstrated experimentally. So for example, the proton gradient can be artificially introduced. ATP is synthesized if ATP synthase, ATP, and inorganic phosphate are present. Okay? So literally, you can see pH 3.8. It's the thylakoid here. It has ATP synthase, right? Reaction, ATP formation. Look how much ATP was made in the acidic solution. Good. I would pause it here, answer these questions, study this experiment, talk about it in class. So the role of oxygen, most of ATP is formed by oxidative phosphorylation, transferring of electrons from NADH to oxygen, which is due to the reoxidation of NADH. So some bacteria and archaea use other electron acceptors, sometimes iron, even uranium, possibly helpful in environmental cleanup. So here's the big pictures, right? This is all of it together. 1, 2, 3, and 4. The goal is to make ATP. Need to know the roles of NADH and FADH2. Need to know the 4 different steps: glycolysis, prep reaction or pyruvate oxidation, Krebs cycle, oxidative phosphorylation, with ETC. There's the electron transport chain. Okay. All right. And then here's another couple of questions, okay, that I want you to pause and think about, all right? What would happen if cyanide was present? Cyanide reversibly binds to the ferric ion cytochrome oxidase 3 within the mitochondria. Cytochrome C right there. Okay. What would happen? What would happen if oxygen was removed? What would happen if you added an uncoupling protein, a mitochondrial intermembrane protein that is a regulated proton channel, which means it dissipates or nullifies the proton gradient? What would happen? Okay. Thermogenin, called uncoupling protein by its discoverers and now known as uncoupling protein 1. It's a mitochondrial carrier protein found in brown adipose tissue. Adipose meaning fat. It is used to generate heat by non-shivering thermogenesis. Creation of heat, not shivering though. And makes a quantitatively important contribution to countering heat loss in babies, which would otherwise occur due to their high surface area to volume ratio. I would like for you to look at the picture and think about how is it creating heat, similar to the question I just asked you, okay? All right, here we go. The last PowerPoint for cellular respiration. If you're still watching this, you can write this phrase: on the cellular respiration test and get a little extra credit for watching this entire video. Now, I don't want you to share with anybody because, hey, you did the work and you should reap the benefits. Don't enable laziness, but encourage others to work hard, okay? So here's the phrase. Let me come up with a phrase. Light is energy, right? Light is a form of energy. We've been talking about energy. Heard this morning in church service, God is light, and in him there is no darkness at all. So that's the phrase. God is light, coming up on Christmas, right? And in him there is no darkness at all. God's character is perfect. It's amazing. One more time. God is light, and in him there is no darkness at all. All right, here we go. Metabolic pathways are linked. There is an interchange of molecules into and out of the pathways for synthesis and breakdown. Carbon skeletons can enter either catabolic breaking down or anabolic building up. These relationships comprise a metabolic system. Okay, so we're going to talk about metabolism a little bit, right? So when you eat this, which, again, not encouraging you to do often, but every now and then, and, you know, definitely when it, you know, we could talk about the beer in the background. Talking about alcohol, right? Fermentation. There you go. Made the beer. But as far as ingesting it, we'll talk about alcoholism down the road. Here we go. You're ingesting all 4 macromolecules there, right? Carbohydrates, lipids, nucleic acids, and proteins. A lot of fat. Of course, the fried, trans fat. Fat in the cheese, fat in the meat, fat in the bacon. I think there's also fried onion rings right there. You got a little vegetables to make you feel better. The toast, the bun probably has like butter on it. I mean, it's just, if you're hungry right now, I'm sure this is not causing anything good if you like to eat hamburgers and stuff. But nonetheless, right? If you eat this, I mean, is only the glucose used for food, or is other things used for food? And of course, the other things are used as well. So here's where it can all enter into. So this is what's important, right? Understanding that, like, how do people not, you know, perish? How do they keep making enough ATP if they're not eating carbohydrates? Well, because the intermediates can go into a lot of different ways. So let's take lipids, for example. Lipids are changed into fatty acids, which can go directly into cell-CoA, okay? Starch, rice, potatoes, being broken down into glucose, okay? Proteins. Some amino acids can go directly into the citric acid cycle. Also nucleic acids, possibly directly into it. Amino acids, purines as well. Some amino acids can go into certain steps in glycolysis. So there's multiple different places where these molecules can go into, okay? So polysaccharides are hydrolyzed into glucose. Lipids can break down to fatty acids and glycerol. Fatty acids can be converted into cell-CoA. Proteins are hydrolyzed or broken down using water to amino acids, which can feed into glycolysis. That's important. Or the citric acid cycle. An oxidized gram of fat produces twice as much ATP as an oxidized gram of carbohydrate. Think about hibernating bears. Digestion. Exergonic, you know, digestion, exergon, releasing energy, right? Releasing energy is important. And so, let's take off the writing. There's just another picture here. Amino acids, sugars, fats, even certain purines and pyrimidines can get involved as well. So biosynthesis, we're making, right? Food is used to provide small molecules for building new, right? So what we do is when we eat, we break it down into building blocks. Love Legos. Still probably going to make that start that club. But some small molecules come directly from food. Others are products of glycolysis or the citric acid. So we're building stuff. We're synthesizing. Example is protein synthesis. And so that takes energy, right? Because we're building something. And so amounts of different molecules are maintained at fairly consistent levels in the pool of metabolites, right? This is accomplished by regulation of enzymes, allosteric regulation, talked about that, right? Active site versus another site, or feedback inhibition, which is when the products cancel out the making of them, right? We don't want to make stuff if it's around that's energetically unfavorable. So enzymes can also be regulated by altering the transcription of genes. We haven't talked about that, but controlling if genes are being transcribed and translated is also a big way to regulate if enzymes are being A, even made, or B, being used. And so ATP and reduced coenzymes link catabolism, anabolism, and photosynthesis. Cellular respiration and photosynthesis are linked by their reactants and products. Okay? So in cellular respiration, glucose is oxidized. In photosynthesis, light energy is converted to chemical energy. So we're making carbohydrates here from light. And there they go right there to produce chemical energy. Super important, big theme there as far as biology goes. And so this picture right here, what is this line doing? Is this a line? It's weird. In some form, all cells perform cellular respiration. Making ATP, being able to build stuff with reduced coenzymes and ATP, right? ATP. Those coenzymes, photosynthesis, green plants, algae, some protists, some prokaryotes perform photosynthesis, taking light, right? And all the processes there. Carbohydrates are formed by photosynthesis. Oxygen is released, used by cellular respiration, CO2. Again, plants do both. So here's your last activity. 3 questions, right? So here is the scientific skills exercise I want you to do. We'll talk about it in class. I'll give you some time to work on it in class. Thyroid hormone levels, oxygen consumption rate. Again, we'll think about this down the road. And so you can pause it here and try to practice this before class. Again, I'll give you some time to do that. And then here are some questions as well. Gave you some pictures there. Fatty acids versus carbohydrates. And you can pause this. Hey, if you watched it this far, great job. Super proud of you.